Good afternoon. We are starting this workshop about the research on pharmaceutical technology in Iberoamerica. We have the participation of three speakers from different countries. They will try to show us the situation of the pharmaceutical technology in their countries and their universities at postgraduate studies level and at research level. An example, they'll explain us briefly their current main research line and their achievements. These three selected conferences cover both innovative aspects of pharmaceutical technology as well as other more traditional but alike highly relevant worldwide. Dr. Manuel Córdoba Díaz from the Universidad Complutense de Madrid, Spain, are going to talk us about the research and development of safe and effective phytomedicines as a strategy to promote the economic, social, health and scientific development of regions with an abundance of vegetables with medicinal properties. Dr. Josimar Eloy from the Universidade Federal do Ceará, Fortaleza, Brazil, is going to talk us about the possibilities offered by different types of nanoparticles of targeting systems for anti-tumor drugs, increasing their effectiveness and decreasing their toxicity. And last, Dr. Carlos Tomás Quirino Barreda from the Universidad Autónoma de México-Xochimilco will talk us about the design and development of new and innovative control release systems for topical and transdermal administration of drugs. Professor Quirino also is founder and administrative secretary of the Ibero-American Conference of Faculties of Pharmacy, COIFA. Dr. Córdoba?
Thank you very much. Thank you very much. First of all, I would like to express my gratitude to the organizing committee for giving us the opportunity of presenting our results here for our project about pharmaceutical development of herbal medicinal products in cooperation. It's a cooperation research project between the Complutense University of Madrid and the private university Antonio Guillermo Urrelo from Peru, and also with the collaboration of Carolina Foundation and Universitario Institute for Industrial Pharmacy. The cooperation of the UCM of the Complutense University and the South American universities is very long, and in the field of pharmaceutical technology, we started to collaborate in 2012 with the Universidad Peruana Cayetano Heredia in Master in Pharmaceutical Technology and a diploma in Quality Assurance for ended product. Also in 2013, a Master in Pharmaceutical Science with the University of the Valley of Guatemala, and in 2014 with the private university Antonio Guillermo Urrelo in Peru, materialized in an agreement for academic, scientific, and cultural cooperation. This is the text of the agreement signed by our two chancellors of both universities, And also the start of this agreement was the first starting point from the Universitari Institute for Industrial Pharmacy for the Complutense University and with the Carolina Foundation. This agreement is based on 3 pillars, that is education, research, and transference of technology. The education is materialized through the design and implementation of postgraduate courses. The research is defined as a definition of a research line. We proposed to look for a research line in cooperation in phytotherapy, and the transference of technology is focused on the transference of technology for the design and elaboration of pharmaceutical dosage forms. Everybody knows that in South America there is an exceptional biodiversity which provides us a high potential for the development of phytomedicinal products. It's one of the most promising areas of the planet, and especially in South America is especially promising from my point of view the region of Peru, because in Peru we can find more than 30 climate zones, more than 100 life zones, numerous ecological floors, and a wide variety of ecosystems. This is the situation that provides us a situation in which it is estimated that more than 25,000 plant species can be found, as estimated. And this is because this fact is because in Peru we can find different altitude from the sea level that provides different ecosystems, and it is especially important for us, the ecosystem in Cajamarca, in the region of Cajamarca, because Cajamarca is just in the middle of this altitude, is just about 2,500 or 3,000 meters over the sea. That is the good ecosystem, the ideal ecosystem for some plants like Sambucus peruviana, for example. We focus our attention to Sambucus peruviana because we have a previous experience with the species, the similar species that we have in Europe. That is Sambucus nigra, for example, or Sambucus ebulus, one of our fields of research. The uses in traditional medicine in Europe for Sambucus nigra is as a diaphoretic, in colds with fever, increased sensitivity of sweat glands to thermal stimuli, and also it is interesting, it has promising uses in traditional medicine in hyperuricemia. Sambucus peruviana has been used in traditional medicine in South America for the treatment of respiratory diseases as an hypoglycemic properties when incorporated into the daily diet. Also, it has been observed an anti-inflammatory effect, and hypolipidemic properties may be attributed to flavonoids and tannins, vitamin C and E. That's why the starting point of this project was some preliminary studies made in Peru with infusion from Sambucus peruviana leaves to study the possible antilipemic effect. So the objective of this project is focused on the performance of a formulation, to carry out a formulation study to design and characterize new formulations for all administration of Sambucus peruviana, powder in direct compressible tablets and capsules with potential antioxidant and antilipemic effect. But also, the other main objective is to enhance the teaching and scientific collaboration between both universities, promoting the training of students and young researchers from Peru and familiarize them with the development and design of pharmaceutical formulations. About the material and methods that we present in this project, first of all, we will make preliminary studies with infusion from leaves to see the antilipemic effect, the preliminary study that I have performed in Peru, the characterization of raw materials in terms of polyphenols content, the obtention, how to obtain the powder, the optimization of the milling conditions, the characterization of powders in terms of particle size, water content, and flow properties, and some formulation studies for the obtention of directly compressible tablets. As I mentioned, the first step is the collection and drying of the leaves, pulverization first drying and pulverization to obtain the powder, and then calculating the loss on drying, the water content by loss on drying, the tablet density, flowability, sieving analysis to study the particle size or the mean particle size and the particle size distribution, and the polyphenols content. To study the polyphenols, the content in the concentration of polyphenols, first thing we did is to optimize the process of extraction. We made an extract with all these steps. First, an extraction with a buffer solution in 7.4 of pH, then a subsequent shaking, filtration, centrifugation, and finally stored it in 2 milliliters aliquots, frozen until use. And the polyphenol content was analyzed by Foley Circulation method. About the results, the first results are concerning to the preliminary studies with the infusions from leaves with a potential antilipemic effect. They collected the leaves from Sambucus peruviana plants, they dried it, and they obtained the powder, and studied its possible effect in a population of 73 patients, more or less half male and female. And these are the preliminary results that they have obtained, with a promising effect in the decrease of total cholesterol, of LDL cholesterol. So from this beginning, we have started with the characterization of raw materials, studying the polyphenol content. The first thing that we have done, we have studied is the optimization of the milling conditions, just to find out if the different milling conditions could affect the composition of the final powder in terms of polyphenol content by Foley Circulation method in terms of gallic acid equivalent. We studied the milling in a micronizer, a pulse miller, and a constant phase and a constant rate miller. This is the calibration curve that we obtained. These are the first experiments for Foley Circulation. And with that, we have obtained these preliminary studies in which we can see that the composition of polyphenol in terms of gallic acid equivalent per grams of sample is slightly, we found a slightly decrease in Sambucus nigra in comparison with Sambucus peruviana, around 10%. And also we have found less gallic acid equivalent in the dried samples in comparison to the fresh ones. When we studied the different milling conditions, we found also a slightly lower content in the Sambucus nigra samples. Then we started with the first preformulations of directly compressible tablets. We started with three direct compression excipients, and also we tried some formulations with some little proofs with a little amount with by wet granulation with hydroxypropyl cellulose and polyvinyl pyrrolidone as a glutant. And then in this first step, this cooperation was materialized through the consecution of two courses as teaching activities in Peru, taught by professors from the Faculty of Pharmacy of the Complutense University of Madrid, from our department. About the characterization of powders, first thing that we studied was water content, and we found that as an average, the water content was around 6 or 7%, what is quite a big amount of water. And maybe it has something to do with the rheological properties. We have found that the rheological properties of these powders are not very good. The flow rate we couldn't calculate the flow rate because there was no flow in our system, and also we found very high values for Carr index, Hassner ratio, and about the particle size, we found not very wide population with a particle size around 0.2, 0.1 millimeters. So maybe the amount of water is responsible for these rheological properties, these poor rheological properties. About tablets, once we started with the first tablet, we obtained this was the best formulation that we obtained. We could obtain tablets of around 540 milligrams with a hardness value of about 75 newtons and a disintegration time that meets the pharmacopoeia European pharmacopoeia requirements. We also started to study the dissolution profiles of these directly compressible tablets. We found that the dissolution profile in water provide us 100% of release in terms of polyphenol in around 3 hours or even 2.5 hours. But the situation was dramatically different when we changed the dissolution medium from pH 1.2 from 0.1 normal hydrochloric acid solution and with a phosphate buffer solution, pH 7.8. If we compare these three profiles, we can see that the profiles are very different, but also we calculated some parameters, modelistic parameters like dissolution efficiency at 5 hours, and we can see a dramatic decrease of the dissolution efficiency when we change to another media because of the modification of the pH and also for the ionic strengths that modify the solubility of the polyphenols. The same conclusion can be reached when we calculate the mean dissolution times, in which there is a big increase when the pH 7.8 and the phosphate buffer solution is used. So, as a conclusions of the results that we have up to this day is that preliminary studies made with 73 patients showed promising antilipemic effect that encourage us to face future studies with more patients and makes necessary the designs of an oral formulation. An analytical method to determine the total polyphenol content, previous extraction from fresh leaves has been developed, implemented and validated for the study of samples for Sambucus nigra and Sambucus peruviana. Polyphenol content was successfully used to determine the integrity of the samples under different milling conditions. It was evidenced that all conditions under study did not modify significantly the content of both species. Also, it was found that Sambucus peruviana showed approximately 10% more content of polyphenols in comparison to the data obtained with Sambucus nigra. Powder samples obtained from both species were pharmacotechnically characterized. It was found that the water content in milled samples was always over 5%, indicating a high hygroscopicity, what can be also responsible for the poor rheological properties. Direct compressible tablets were designed and characterized. The formulation met the requirements for hardness and disintegration time. Concerning the dissolution profile, it was found a clear relation between dissolution rate and pH and ionic strength. Water resulted to be the medium in which the polyphenols were released faster with dissolution efficiencies near 90%. So we could see in our plots. Not only a research, but also a teaching collaboration have been implemented between both universities, UCM and UPAGO. This promising collaboration is still working with future courses in this academic year and on the vision of new students for Peru to familiarize them with pharmacotechnical research and to face new strategies like the future development that we are now starting to face of semi-solid and liquid orally administered formulation. And these are some pictures about the teaching collaboration of our both team from the university from Peru and our university, and also with the support, also it's a time to express our gratitude also for the support of our Department of Pharmaceutics and Food Technology, and of course from the support from the Faculty of Pharmacy of the Complutense University of Madrid. Thank you.
¿No está funcionando?
Parece que no. Y este tampoco.
No está funcionando.
No funciona el ratón del ordenador. No nos ven.
Me había olvidado que ustedes le llaman ratón aquí en España. Nosotros llamamos mouse
Claro.
en Brasil. No funciona el ratón. ¿Ustedes no tienen un laser pointer? ¿Esto es?
Sí, se pone con láser, creo que está arriba. Sí.
¿Eso también cambia los slides?
Sí.
¿Sí?
Voy a por otro ratón, sí. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66, 67, 68, 69, 70, 71, 72, 73, 74, 75, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, 100, 101, 102, 103, 104, 105, 106, 107, 108, 109, 110, 111, 112, 113, 114, 115, 116, 1
estimada. Isso porque é um Apple, hein? Tudo é Apple, hein? Aqui não se supone, não... No, ese no es el mío. No, estaba... Este es el mío, ¿verdad? Sí. No, no es este. Lo había dejado aquí. Pero yo lo había guardado aquí. No, no es ese. No. No está más aquí. Pero yo lo tengo.
¿Alguno de estos también son PowerPoint?
No, pero no ese es el nombre. Lo había dejado aquí, pero ya no está. ¿Dónde lo pongo?
Espera un momentito. Aquí hay un...
Este es el de... Sí, este es el del... El puntero.
Ah, pues entonces no.
Aquí.
Sí, es que si no, no te lo acuerdo. Ahora no entra con el chat.
Qué raro porque lo había puesto aquí. Control. Ahora sí.
Si quieres que ponga mi ordenador, también lo...
No, yo creo que está bien. ¿Puedo quitarlo? Sí. Y ahí lo puedo cambiar por aquí.
Dale a presentación. Mira si funciona. Vale, sí. Ahora te lo ponemos, ahora te lo ponemos.
Ajá.
Ah, vale, bien.
Gracias. Well, good afternoon, everybody. My name is Josimar de Eloy. I am from Federal University of Ceará in Brazil. Sorry for the delay that we had, but let's move on. Well, before I start with the presentation and showing the research that's being conducted at Federal University of Ceará, I would like to show you here we are on the northeast coast of Brazil, and this is the main building of the university, and this is the Department of Pharmacy. These are some pictures of my lab. This is the Faculty of Pharmacy, Odontology, and Nursing. So unlike here, we have 3 different courses at the same faculty. And the Department of Pharmacy is very young if you compare with the reality here in Europe, so 100 years. And in the undergraduate course of pharmacy, we have over 500 students enrolled in the course. And I'm participating in the Pharmaceutical Sciences post-graduation program. And nowadays, we have 83 PhD and master students in different areas of the pharmaceutical sciences. My area is Pharmaceutical Technology. So this is showing some of my research interests. So I work with the development and characterization of drug nanocarriers, so different types of drug nanocarriers, for example, polymeric nanoparticles, lipid nanoparticles such as liposomes, and we study these nanocarriers with different techniques physicochemical techniques. And also we evaluate these nanocarriers for cancer treatment. So basically we test these different nanocarriers that we have been developing in different cancer cell lines. And also we developed some xenograft models for doing the in vivo evaluation. And also we are focusing on targeted nanocarriers. So we modify the surface of these nanocarriers with some ligands for overexpressed receptors on tumor cells. So we are trying to direct or target these nanocarriers to specific cells as an approach to reduce side effects. So this is a slide showing how small these nanocarriers are. So usually they have this size between 1 and 100 or 200 nanometers. So because of this very small size, there are some improved characteristics. So if we compare the nanocarriers with some other entities, for example, here we have atoms, molecules, proteins, bacteria, and cells, we can see that the nanoparticles are more or less the size of some large proteins, and they are much smaller than most cells. So this is an important characteristic that influences the behavior of this type of delivery systems. Also, here we have a variety of these types of carriers. So we can make these nanocarriers with polymers, natural or synthetic polymers. We can have nanospheres. We can also have nanocapsules, polymeric micelles, which is very interesting, are being researched worldwide. Also dendrimers, this branched structure of dendrimers are very interesting for encapsulation of different drugs. And also we have hydrogel nanoparticles. So we have a very high versatility of this type of delivery systems. Also we have the lipid nanoparticles, which are also very different from one another. We have the liposomes, which are the traditional delivery systems. They have been described for over 50 years. So there has been a lot of development in the liposomal research, and actually nowadays we have some products that are already approved in the market, especially for cancer treatment, but we also have other diseases that can be treated with liposomes. We have also, so we can here see the lipid bilayer, so liposomes are formed with phospholipids, and we can encapsulate here hydrophobic molecules, and in the inner compartment here, which is aqueous, we can encapsulate hydrophilic molecules. So this is an important advantage of liposomes because we can deal with drugs with different polarities. Also, we have solid lipid nanoparticles, which usually have better colloidal stability. This is an important issue when we talk about delivery systems. We also have phospholipid micelles, as we have polymeric micelles. We have the same structure. And last but not least important, we also have the metal and inorganic nanocarriers, so made with different materials such as iron, gold, silver, and they are used especially for diagnosis. For example, gold nanoparticles is used for plasmonic therapy, for heat generation, which is important for cancer diagnosis. And also these systems we can link on the surface. We can link also drugs, and the systems can be used as theranostic tools, so both for diagnosis and treatment, which is very exciting. And here, just to show you some of the market, because this area is really growing fastly. And this market, it's not only in health. We have the nanoparticles used in different areas of technology. And here we have the nanoparticles itself. They occupy a very large proportion of these nanostructures. And we have here liposomes in green. So a lot of these nanocarriers are liposomes, and most of them are used in the medical field. So here on the right, we have the application in medicine. Just a few examples, we have much more. The first one, which was approved by the FDA agency, is the Doxil It's a liposomal, a pegylated liposomal formulation containing doxorubicin. So this is used to treat a wide variety of cancer types. And which is very important and interesting about Toxil is that because of the incorporation of doxorubicin in this formulation, it was observed in the clinical settings a reduction in side effects. For example, the cardiotoxicity, which is something that's very concerning doctors when treating patients. We also have a version of this liposomal formulation for doxorubicin delivery, which is Myoset. And comparing these two ones, Myoset is non-pegylated. And because of this, the release is faster. So here we have a formulation developed for faster release. And here with Toxil, we have a formulation developed for more sustained release. And not only we have liposomes, but we also have other carriers. For example, here we have Abraxane, which is a polymeric nanoparticle made of albumin. Albumin can be found very easily, so this formulation is cheaper. And to deliver paclitaxel, which is also used to treat a lot of cancer types. So this area is increasing a lot, and nowadays we have many clinical trials being conducted, and we expect that in the near future we can expand each more the use of nanoparticles for cancer treatment and also diagnosis. And what are their functions? So I'm talking about nanocarriers. Why are they so important? First, because they're able to control drug release, so we can have a sustained, slower release. And also we can improve how these nanoparticles will reach the target. So we are talking about many molecules that have poor bioavailability. So when we encapsulate them within a nanocarrier, we can improve how this drug is delivered to the target, for example, to the cancer cell. And with this, we can have decreased side effects because the nanocarrier will be delivered to the cancer tumor and not to the brain, not to the heart, not to other tissues. So this is very important. And some of these important characteristics of nanocarriers, as I said, they have a very small particle size. And here we can represent. So because of this, as a consequence, there is a very high increase of surface area. So usually because of this, we have an improvement in the solubility. And many of these drugs are lipophilic. And because they are lipophilic, they have poor bioavailability. So when we encapsulate in unstructured delivery systems, we can observe an improvement of solubility. Also, we can see an improvement of stability. Because it's encapsulated, it can be protected from the environment, from oxygen, from water, from factors that will affect the stability of these drugs. And also, we can have the controlled release. Because, of course, for this drug can exert the action, it has to be released to reach the target. And in this figure, we can see the conventional administration of drug. It's just a general example. So we can have different dosages, and when the patient takes the first dosage, usually we have the absorption of these drugs, and we can reach some toxic plasmatic concentrations. And of course, there is the clearance, the metabolism occurs, and then we can have some subtherapeutic valleys. So with the normal, the regular, general administration of drugs, we can have these toxic valleys, peaks, and subtherapeutic valleys. And when we offer the nanoparticles, when we administrate the nanoparticles, because the release is sustained and slow, we can avoid the toxicity and the subtherapeutic plasmatic level. So this is one of the biggest advantages. And also, it's important that once the nanoparticle is administered, it is maintained in the blood circulation without being fastly cleared by macrophages, for example. So we can pegylate this surface. We can use PEG, polyethylene glycol, and modify this surface. So we can create a hydrophilic barrier which can prevent the fast uptake by macrophage, by circulating macrophage. So we can have a prolonged circulation of these nanoparticles. And also, the tumor is developing really fast, adapted for this reason, and it is taking up a lot of nutrients, oxygen. So all the microenvironment around the tumor is developed in this context of offering nutrients to the tumor. So the vessels, the blood vessels around this microenvironment are fenestrated, which means that they have small holes. So the nanoparticles, when circulating, they can extravasate through these small holes, and they can concentrate. The drainage, the lymphatic drainage here in the tumor is very poor. So the nanoparticles extravasate and they accumulate here in the tumor. So this is known as the EPR effect, enhanced permeation and retention effect. So the nanoparticles are passively directed to the tumors. And what I'm researching right now is how we can actively target these nanoparticles to the tumors. Before I speak about the targeted delivery systems, let's focus a little bit more on the structure of liposomes. Here we have unilamellar liposomes and also multilamellar liposomes formed with these phospholipids. So they have a lot of advantages, such as biosafety, because they are formed with phospholipids, membrane phospholipids. They are able to encapsulate both hydrophilic and hydrophobic drugs. They protect against degradation, are able to perform sustained release, and as a consequence, to reduce side effects of the encapsulated drug, of course. So, as I said, nanoparticles can be passively accumulated at the tumors because of the EPR effect. But we can go further. We can modify these nanoparticles with monoclonal antibodies, peptides, folate, transferrin, carbohydrates, aptamers, so different molecules that can be recognized by the tumor cells. So, of course, we know we need a biochemical characterization of the target. For example, the cancer cell, the breast cancer, the lung cancer cell. We not only have one type of breast cancer cell, we have many types of cancer cells, and we have to know which receptors are overexpressed in this particular cell line. So we can design a nanoparticle with the ligand for this receptor as an approach to increase the uptake, the cellular uptake of this nanoparticle. So for example, for breast cancer, for HER2 is a receptor that's overly expressed in a variety of breast cancer cells and are usually associated with poor prognosis. So we can link the monoclonal antibody to the liposome, and so the liposome or the immunoliposome will be increased uptake by the cell. And also, we have some clinical trials already. We don't have a product in the market, these targeted or functionalized nanoparticles. We don't have them yet in the market, but we have some promise. We have some clinical trials that were conducted or are being conducted. For example, transferrin is related to iron uptake, so the cancer cell needs a lot of iron. So some cancer cells have increased iron receptor overexpression. So transferrin receptor targeted oxaliplatin liposome, phase 1 and phase 2 studies. We have transferrin receptor targeted siRNA polymeric nanoparticles. There is an increase in interest in antisense oligonucleotide, siRNA, as it represents maybe a future in medicine, as Dr. Philip Sharp was mentioning in his lecture. We have a monoclonal antibody targeted doxorubicin liposome, so an immunoliposome, a phase 2 study right now is recruiting patients for this study. It's been conducted here in Europe, the Swiss Group for Clinical Research. And we also have antibody-targeted doxorubicin liposomes, but not the whole antibody, with the fragment, a small fragment of this antibody, only with the portion that does the recognition of the cell. And now, in the last part of my presentation, I'm going to show you some research papers that were conducted by my group or in collaboration with other groups. All of them have this in common, that the formulations, the nanocarriers, different types of nanocarriers, were functionalized, so they were targeted with some moieties for improved selective delivery. So one of our interests is the skin delivery. We know that topical delivery of some drugs is very promising and very interesting with many advantages. However, the skin represents a barrier for drug penetration, for drug delivery. So we need to use some absorption enhancers, and pharmaceutical technology using nanocarriers offers an interesting approach to overcome the skin barrier. So we published this paper, liquid crystalline nanodispersions functionalized with cell-penetrating peptides, TAT and penetrating are the two peptides that we studied, for topical delivery of siRNA, a proposal for silencing pro-inflammatory cytokines in cutaneous diseases. So here we explored the TNF-alpha, which is involved in a lot of inflammatory cutaneous diseases. And this figure shows in polarized microscopy the behavior of these formulations, these liquid crystalline formulations. This is the hexagonal phase, which is interesting for skin delivery. And we prepared them with monolein, oleic acid, and also using the peptides, TAT or penetrating. Here we performed the skin delivery in porcine skin. So we did these in vitro studies comparing the different groups. And we can see the marker here for drug penetration in green. We can see the color here in green. And we have a better delivery, especially for the inner layers of the skin, which is more important, the vial epidermis. We had, so the best result here in terms of fluorescence with the formulation functionalized with the TAT peptide, so the cell-penetrating peptide. And we observed a correlation with the in vivo result. Here we have, it was done with ELISA study. We did in vivo skin suppression of TNF-alpha, and the formulation that was able to better suppress the TNF-alpha was the formulation here that was functionalized with TAT peptide. So a nanodispersion, a liquid crystalline nanodispersion in the hexagonal phase modified with TAT peptide was able to suppress the TNF-alpha concentration. So we were very excited with this study. It was published 2 years ago. And also, this is a different type of targeting now for breast cancer. And in breast cancer, our focus was to target the HER2 protein, which is a transmembrane receptor in some breast cancer cell lines. And for that, we performed a covalent binding between the antibody here, which is trastuzumab, which is currently used to treat some types of breast cancer. It's in the clinic. We linked them in the liposome with this chemical group, maleimide. So when we use the Trout's reagent to modify the antibody, so the antibody is modified, it's against the sulfhydryl molecule. And the sulfhydryl molecule is reactive with the maleimide group. So we have here the covalent bond, and then we have the formation of the immunoliposome through this thioether bond here. So we have the immunoliposome. And we explored this immunoliposome, and we evaluated in this paper rapamycin-loaded immunoliposome with trastuzumab, a strategy to enhance cytotoxicity to HER2-positive breast cancer cells. So we observed the synergistic effect between the antibody trastuzumab and rapamycin, which is a drug candidate to be used in breast cancer treatment. And we continued this study. We published last year in European Journal of Pharmaceuticals and Biopharmaceutics. The paper is anti-HER2 immunoliposome for co-delivery of paclitaxel, which is commonly used for breast cancer treatment. So we made an association with rapamycin for breast cancer therapy. And here I'm showing the characterization of these immunoliposomes. This is the microscopy picture, and also the electrophoresis picture showing the presence of the antibody. We have here the heavy chain of the antibody and the light chain of the antibody. And in this table, I'm summarizing the results of functionalization efficiency, encapsulation efficiency of rapamycin, paclitaxel, and also the nanometric particle size, the polydispersity, and the zeta potential. We performed the evaluation in cell culture using the MTT assay, and we observed a synergistic effect between paclitaxel and rapamycin in presence of immunoliposomes, which means in presence of the antibody. We performed this evaluation in HER2 negative cell lines, 41 cell lines, and HER2 positive cell lines, SK-BR3 cell lines. So we only observed this synergistic effect when we had the HER2 positive cell lines as expected. And here we have some confocal results using both the negative, the receptor negative cell line and the receptor positive cell line. And in green here, we can see the fluorescence of the nanocarrier in the cytoplasm of the cells, so showing an improved uptake of this nanoparticle when decorated, when functionalized with the antibody in presence incubated with the HER2 positive cell line. And in vivo, we had the confirmation of this in vitro results. And here we have the xenograft model for breast cancer. We injected breast cancer cells in nude mice, and so they developed the breast cancers, and we performed the administration of different groups. And here in black, we have the immunoliposome group. So we can observe here this, in the X, here we can see the time after progression of the breast cancer and the tumor volume. So the group treated with immunoliposome, it had the lowest growth of this tumor. So they were the best group. And at the end of the study, we extracted these tumors, and what we observed is that these tumors were really smaller when they were treated with the immunoliposomes compared to the other groups. And we also performed some histological analysis, and what we could observe is that for these tumors treated with the immunoliposome, there was a decrease in the mitosis index. So the cells were dividing more slowly. That is what we observed. And also, my last example is another target, not now for breast cancer, but for skin squamous carcinoma cells, which overexpress another type of receptor, which is the EGFR. So in this approach, we tried to direct the immunoliposome to EGFR. And how did we do that? We linked with that same reaction that I showed you before. We linked another antibody, now cetuximab. We linked this in the liposome. And the liposome contained 5-FU, which is a drug commonly used for this type of treatment. And we observed here the synergistic effect between cetuximab, the antibody, and 5-FU, the small drug. We performed this study in A421 cell lines, which are EGFR positive cell lines, and we observed this synergistic effect when we incubated the immunoliposome containing the 5-FU drug. And we moved forward with this first report. We recently published in Journal of Controlled Release. This paper, Skin Cancer Treatment Effectiveness, is improved by iontophoresis of EGFR-targeted liposomes containing 5-FU compared with subcutaneous injection. So we compare two different routes of administration, the topical route and the subcutaneous route. And to aid skin delivery in the topical treatment, we use a technique called iontophoresis. So this is the experimental setup here, a demonstration. Iontophoresis uses electric current to force drug penetration through the skin. So the electric current reduces this barrier effect, and so we can improve the drug delivery through the skin. And here we can see the effects of the liposome and the immunoliposome using the cell lines, the EGFR negative and positive cell lines. And what we observed is that the immunoliposome had in green, here it's evident, an improved uptake of the formulation. So we could see the interaction between the receptor and the ligand. Here, we also checked the same with flow cytometry, not only with confocal, but also with flow cytometry. We could confirm that the uptake was increased. And also, we did some other experiments, Western blotting, where we could confirm that EGFR was present in this uptake. So the liposomal uptake was mediated between the interaction of EGF, the ligand, and EGFR, the receptor. So we proved this with Western. And also, we quantified the amount of drug that were delivered through the skin in different layers using iontophoresis or not using iontophoresis. And what we observed that when we quantified 5-FU in different skin layers, and what we saw is that iontophoresis really helped the skin penetration, especially when 5-FU was encapsulated in immunoliposomes. And what's the reason? Because the epidermis, especially in deeper layers, they have increased EGFR receptors, so the immunoliposome is able to target these receptors when it reaches deeper layers of the skin. So we were able to quantify much more 5-FU when we used the immunoliposome. And in vivo, we also performed the xenograft study. And here we have the comparison between the two administration routes, the subcutaneous here in B, and iontophoretic delivery. And what we could observe is that we had an even better efficacy result when we used the iontophoretic delivery, when we used electric current. Here in green, it's a little hard to see, but we have the immunoliposomes. And the tumors were way smaller when we used immunoliposomes treated topically using iontophoresis. So that was the main result that we had. And here we also extracted the tumors at the end of the experiment, and we confirmed these smaller tumors when they were treated with the immunoliposomes. And also we published some review papers in the field with hydrophilic drugs, with antisense oligonucleotides, siRNA, all talking about targeted delivery. Also some book chapters, and this protocol for conjugation, for covalent conjugation, we published the preparation of immunoliposomes by direct coupling of antibody based on a thioether bond. This is Fortaleza in the northeast coast, always warm, never cold. In Brazil, always sunny, it never rains. And also thank the universities. I work in collaboration with University of São Paulo, São Paulo State University, Ohio State University. And this is my team currently. And thank you very much.
Creo que ya. 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 Good afternoon. Before I begin my speech, I would like to thank Dr. Anabel Torres for inviting me to participate in this workshop to share with such a select panel and this audience part of the research we do in the molecular pharmaceutics and drug delivery laboratory in the Universidad Autónoma Metropolitana, Campus Xochimilco, located at south of Mexico City. Of course, I shall present the results from a collaborative and multidisciplinary work team with peers from other laboratories in my university and other institutions in Mexico City. In particular way, I also want to mention that in this research, as in all the projects that we developed in our laboratory, participating in major way students of degree level pharmacy as well as of master in pharmaceutical science and the PhD program in biological and health science, and from other disciplines such as dentistry and industrial design. And the agenda that I prepare for this panel is this. First, I would like to remember some important aspects about the skin that explain why the topical and transdermal drug administration via is as successful nowadays as Dr. Eloy tells all. In second moment, I shall introduce transdermal drug administration via with abording aspects about liquid crystalline state of the matter and how about this related with the lipids that exist in their skin. And finally, I will present some physicochemical results obtained with exosomes system containing ketoconazole as a liquid crystalline dispersion system with topical and transdermal potential as drug delivery system. Well, first, in this slide, I will share with you about the skin and cellular membrane lipoprotein and liquid crystalline structures that controlling permeation. And it's important to say that the integument system of vertebrates is a complex and unique organ system. In humans, it is not only the largest and one of the most readily accepted organ in the body. It covers suffers approximately 2 square meters in an adult, and it receives about 1/3 of the blood circulating through the body. But in terms of functions performed, the skin outweighs any other organ because it covers functions like sensorial, physical, chemical, immune, pathogen, UV radiation, and free radicals defenses. It also participates in thermoregulations and performs very important endocrine functions for health, so vitamin D synthesis and peripheral conversion of hormone, as well as their significant role in human reproduction. In this case, I'm sure everybody in this room knows what about I'm trying to say, isn't it? why does not knowing the important role of the skin when we have visualized a woman or a man, it depends on preference, of course, or when unconsciously I inhale the aroma they emanate. We will feel then something special or difficult to explain, of course, but actually it is happening one expression of biochemical phenomena of our skin through secondary sexual characteristics and express emotions. Because of this, the skin has not only been a target for the pharmaceutical industry, but also for cosmetics, personal care, and fashion business. In this slide, we can show what is the structure and the functions, and the important is the stratum corneum, why the corneocytes are surrounded by lipids, and this is the main structure responsible the action and the permeation in the skin. In this slide, I try to show you why the topical system are so important nowadays, because as you see, in nanomedicines, after intravenous systems, topical administration is very important for drug release. 52% of nanotechnological systems are based in these kind of products. This slide explains the important to differentiate or clarify between topical, transdermal, and cutaneous formulation activity. Topical formulations are products applied to the skin surface that may penetrate to some extent in the skin layers. This is the skin, and we can see three main routes to delivery of drugs. It's the transcellular route, intercellular routes, and follicular routes. So after the application, the effects expected with the products help us for the treatment skin disorders as acne, dermatitis, cutaneous inflammatory disease, and others. On the other hand, we need, or we want to the drugs penetrate and need absorbed in the blood, and for that, transdermal composition are designed to release drug substances in this must permeate through the skin and enter to the systemic circulation, ensuring that significant concentration of drug are absorbed to produce effective plasma concentration. In some cases, design is not only to ensure drug permeation, but also drug targeted to body regions close to the action site, where a regional effect is expected, for example, in the muscles, blood vessels, and articulations. It's important to say, too, the term continuous absorption is applied to characterize the sum of the amounts of drug that penetrate and permeate the skin. Nowadays, topical administration is also performed for drug targeting, as shared Dr. Eloy with us, and to the action site in the body, reducing the systemic side effects. The administration of drugs through the skin also useful for drug substances with irregular oral absorption and low bioavailability, and for drugs with increased first-pass metabolism and short plasma half-life times. That explains why many cosmetic, nutraceuticals, and pharmaceutical dosage forms and nanopharmaceutical products commercialized or developed are lipid-based. In this slide, we can see the composition and the lipids surrounding the corneocytes, and they represent the intercellular routes, the main important route to drug deliveries. Composition is ceramides, fatty free acids, and cholesterol and water. And it's important to explain these lipids are bilayer, and they suffer transitions of phase depending on the temperature, thickness, and the profundity and depth, please. These phase transitions are reversible and exist mesophasic changing in soil health structure as lamellar, exosomic, orthorhombic, and cubosomic systems. Other important in this slide is explained to keratinocytes, membrane lipoproteinic structure have a cubosomic phase, and membrane is a kind of liquid crystal system. The stratum corneum arrangement is analogous to wall bricks and mortar. And in that model, corneocytes represents the bricks, and the intercellular lipids represent the mortar, where lipids form several bilayers, as I said before, surrounding the corneocytes. It's important to mention that lipids compounding the mortar in the epidermis, and particularly in the stratum corneum, forming amphiphilic lipids bilayers that could adopt different liquid crystalline phases, and in this layer is continuously modified depending on factors as temperature and where these lipids are temporarily located, so they suffer, as I said before, soil health reversible phase transitions. In a similar way, when the lipoproteins of corneocyte membrane are moist, they could adopt a cubosomic liquid crystalline structure favoring permeation. In this slide, we can observe what I mentioned. previously following the model in the skin as a well-structured brick and mortar. And the lipids adopt this bilayer form, and their structure we can see form lamellar or hexagonal or orthorhombic faces. That explains why the liquid crystal system are now using for drug delivery systems. In this graphic, we can see the depending of the depth in the stratum corneum and the temperature exists in our skin this different liquid crystalline system. We can exist the lamellar phase or hexagonal or orthorhombic depending the temperature and the depth of the skin. Other factors affecting the this performance in the skin are related with the cell membrane and their composition with lipoproteins. And we know the cell membrane as keratinocytes and other cells in our body have this kind of phase structure like liquid crystalline systems. They exist with lipoprotein and other amphiphilic molecules exist in bilayers. When temperature is increased or the number of insaturation in phospholipid chains, we can observe the flow is higher, and when the length of this lipid chains is increased, the flow is and viscosity the flow is low, and the viscosity is higher. It seems occurs with the cholesterol is increased, and exists different mechanisms that explain this situation, and depending of these factors, we can continuous the skin is changing in reversible form between sol, liquid crystal, and gel structure. Other important factor is related with the charge that exists in cell membranes. And they obey to the lipids, and it's usual that membrane lipids have negative charge. And for that, macroions, for example, with calcium, accoupled or complexed with amphiphilic molecules or other molecules as peptides affect this discharge on the cell structure, membrane cell structure, and changing this structure and occur segregation or phase separation. Well, with this background, and now we are talking about the liquid crystals and as a nanotechnologic lipid-based alternative for drug delivery system. And Dr. Eloy showed us an example before. In this case, it's important to say pharmaceutical, nutraceutical, and cosmeceutical molecules, whatever its source, plants, animals, minerals, substance, or biomolecules product, or the biotechnology, most of them are poorly soluble. This reason and their high permeability limits their absorption, and for that, nowadays, lipid-based colloidal systems or nanosomes, as we can see before, have innovative drug delivery systems. They are prepared with isotropic mixtures of oil, large and short chain amphiphilic lipids, and the poorly water-soluble drug. And then the first phase is mixing oils with water and polymeric solutions, and the result is micro or nano emulsions, another system including liquid crystalline systems. Other liquid crystalline systems are obtained when we mix oils and waxy substances and heating and mixing this mix. But the important of this is that they can be added to or constitute different dosage forms as capsules and tablets. We can mix these liquid crystal systems with solid particles and form this kind of products. This is a scheme how in the Universidad Autónoma Metropolitana produce these liquid crystalline dispersions. And first mix, as I said, the lipid mixture, for example, with amphiphilic molecules like lauroglycol, PCol, and a polymeric stabilizer, as poloxamer. After this mixture is mixed with water, and we apply temperature, and finally apply ultrasonic cycles. This is we can observe the product as this process, but we need to characterize that, and after obtain, the first is determination particle size. In this case, like using light scattering, and we could prove this is the nanometric size. After that, we use transmission electronic microscopy with negative staining technique, and we can show the structure, and in this case, we obtain these hexagonal liquid crystalline systems. And if we remember, this kind of liquid crystalline system is similar to the lipids surrounding corneocytes. It's important to say too, each system is different in physicochemical properties. Exo, orthorhombic, and cubic systems are different as the structure. We know exosomes and cubosomes. These are dispersion of these primary systems. These are viscose, and these are pseudo-Newtonians. Now, the last part of my presentation is about characterization of exosomes, and I will present some physicochemical properties obtained with these systems loaded with ketoconazole. Our hypothesis was exosomes as lyotropic liquid crystalline dispersion system loaded with different drugs, including ketoconazoles, are able controlling drug release, diffusion and retention in membranes used as a model in vitro diffusion and permeation. This is examples of this system loaded with different molecules with different log P and other properties as molecular mass, and we can observe in this slide all of these have the exosomal form observed with MET. Yes, and it's important to say too in this slide, observe systems loading with ketoconazole. We obtain a zeta potential positive, and a placebo have a negative charge. The particle size obtained more or less is the same, and after that we study drug release using this dialysis membrane. And what is important of potential effect is in other diffusion studies with positive charge have shown a higher diffusion compared with negative is better through the pore air skin. This is a thermal behavior or system. It was a stability over the time, and at different temperatures, the system maintained this stability, and this stability is reversible. That is important because that occurs in the skin, with the lipids in the skin. Finally, we can see, unlike the commercial product, in this case, the liquid crystalline system with ketoconazole release ketoconazole in the stratum between the first minutes, and it remains during various hours, as we can see in this case. This is the liquid crystalline systems. It releases ketoconazole practically when it's deposited on the skin, and this is the case of the cream, the commercial cream. Finally, in vitro activity against Candida albicans demonstrate the potencies of ketoconazole is increased when it is part of this kind of nanostructured system. So the mix of ketoconazoles against Candida albicans results in a higher activity, as you see in this slide. So that's the importance of obtain this kind of liquid crystalline systems, and explain why these systems retain it in the skin, and is stable. The stability is the same as occurs with the lipids in the skin. Well, that is all that I will share you, and thank you very much.
Well, thank you very much to our speakers for the very interesting conferences. They have shown us 3 examples of the research in Iberoamerica, and well, we don't have enough time for nothing. But I want to name at least another groups from other countries, different groups that have presented here in this Congress. Another works very, very interesting. For example, the group of Dr. Gracia from the University of Monterrey in Mexico, and the group of the Dr. Oliveira from Argentina, that also very interesting works about pharmaceutical technology from the Universidad Central de Ecuador, and another interesting works from Panama and Puerto Rico. I invite you to see more in depth all these works during the Congress. Thank you very much, everyone, for your participation, for your attention.